Hello, my name is Isabelle Townsend from the University of Southern Mississippi, and my research was on biochemical and computational characterization of small regulatory RNAs in Crassostrea virginica, the eastern oyster. So let's start off with some background information. RNA interference, or RNAi, is the process of short, non-coding RNAs associated with Argonaute proteins that target and silence messenger RNA strands before translation can occur. These regulate at the transcriptional and post-transcriptional levels to prevent gene expression. Argonaute proteins can come in two different clades, Argo or Pee-wee clades. They're found in both prokaryotes and eukaryotes, and their role in gene silencing is highly conserved. They basically work by binding to the guide strand, recognizing a target, then there is an annealing phase, and is followed by a target cleavage phase. There are 4 domains to each Argonaute protein. The Pee-wee domain is the active site, and the N-terminal, mid, and pass domains all work to anchor the guide strand and messenger RNA strand, and then unwind the target according to the guide strand. So there are 3 classes of small RNA, microRNA, short interfering RNA, and Pee-wee interacting RNA. They're each unique in their biogenesis and their silencing mechanisms. I'll start with microRNA, or miRNA, because these are the most well-known and the most highly conserved. These short interfering RNAs are about 21 to 22 nucleotides long, and their biogenesis is relatively simple. They are transcribed from the genome into primary miRNA structures that are hairpin structures that are then cleaved by drosha and a binding protein. That is then exported into the cytoplasm, processed by dicer into duplexes with 2 nucleotide overhangs, and then those duplexes are loaded into the RNA-induced silencing complex, or RISC, along with an Argonaute protein in the Argo clade. The less stable passenger strand is then removed, and the mature microRNA, which is about 20 nucleotides long, can identify several different messenger RNA strands. The silencing mechanism then is the miRNA along with the RISC complex, targeting and binding to these messenger RNAs and cleaving them before translation can occur. Next we have short interfering RNA, or siRNA. These are also about 21 to 24 nucleotides long, and they are similar to some extent to microRNA, but they have a distinct biogenesis and silencing mechanism. So these RNAs are derived from double-stranded RNA precursors, and these can be from viruses or other transcript products. Dicer then processes them into siRNA duplexes. That is then loaded into the RISC complex, and the passenger strand is removed. The silencing mechanism from there is targeting the messenger RNA that overlaps with the guide strand and disrupting the messenger RNA strand to prevent translation from occurring. Lastly, we have the Pee-wee interacting RNA, or piRNAs. These are distinct because they are not associated with the Argo clade, but instead the Pee-wee clade, and they work on the germline to protect it from transposable elements. Their biogenesis is also unique, and it can occur in one of 2 ways. The first is being transcribed from piRNA clusters in the nucleus. That is then processed by zucchini to form primary piRNAs. That can then enter the ping-pong cycle, which is a self-amplifying cycle. The primary piRNA can be loaded into a Pee-wee paralog that can identify a transposable element complementary to the guide strand. From there, the transposable element can be cleaved and then used to generate novel piRNAs specific to that transposable element. And this amplifies a lot of piRNAs unique to that transposable element. These piRNAs also have a unique 10 nucleotide overhang and a preference for uracil at the 5' end. PiRNA can then go on to be loaded into either Pee-wee or Argogene to silence their targets. All of these are well documented in model organisms such as Drosophila. have been documented in the other oyster species, Crassostrea gigas. The residues were shown to be prevalent in the gonads, gills, and muzzle of the oyster. In order to categorize the small RNA in C. virginica, we used cephalus beads to separate the unbound RNA from the argonaut-bound RNA via strong basic anion exchange chromatography. Two extraction methods were used: a total RNA extraction with trizol that served as our control, and a selective extraction using the cephalus beads, with samples taken from the gills and the gonads of live oysters. The results of these extractions were then put through a gel electrophoresis using a polyacrylamide gel, where we compared the control total RNA extractions to the extractions done with the beads and the gills and the gonads. The best of these results were then sequenced, and SAM tools and bed tools were used to organize these reads by lengths and then converted into readable files that we then were able to graph. Gel electrophoresis showed that gill samples and gonad samples tended to have the best results, and so those were the ones that ended up being used for sequencing. The data then showed that in the control, both piRNA and smaller micro and short interfering RNAs were found in the oysters, but in the gills and in the gonads for selective bead extractions, only the shorter reads were seen, with a peak of about 5 million residues in the gonads. Compared to the control, which did show piRNAs as existing in our oysters, and compared to previous research done in C. gigas, this was deemed a negative result, and not a true reflection of the population of small RNAs in our oyster. We know from previous research that anion exchange chromatography has worked in other non-model organisms such as spider mites, and so our negative response was deemed an issue with the salinity due to our oysters that interfered with the anion exchange. This higher salinity would interfere with the purification that the beads were attempted to do due to its interference with the anion exchange. So one potential solution to this could be to dilute the binding buffer that we were using to neutralize it, which would then allow the beads to produce a more accurate depiction of the small RNA populations in our oyster. Given the improvements to the extraction method that we used, further characterization of these small RNAs can be done in our non-model organism. In this study, we focused more on size discrepancies between mi and siRNA compared to the longer piRNAs. Other ways of characterizing small RNAs could be through overhangs and through nucleotide content in the small RNAs. For example, piRNAs resulting from the ping-pong cycle have a characteristic 10 nucleotide overhang and a preference for uracil at the 5' end. That could be used as an identifier for characterization. As well, microRNAs, duplexes, have a 2 nucleotide overhang that can also be used to characterize. In addition, the improvements to this extraction technique can assist in other studies using other non-model organisms, which can add to our understanding of how these small RNA classes are created, how they work in RNA interference, and can add to our small RNA databases such as Mirbase that focuses on microRNAs. That concludes my presentation. Thank you so much.